Welcome to today's deep dive. We are opening up a groundbreaking June 2026 paper from Nature Biotechnology today.
Yeah, it's a really exciting one.
And our mission for you today is to completely unpack a new piece of technology called Perturb-DBiT.
Right, which is quite the acronym, but we'll get into it.
To really grasp why this matters, think about how we usually study a disease like, cancer. We tend to break it down to its absolute most basic parts.
Like trying to understand the whole by looking at the pieces.
Exactly. You take a piece of a tumor, you put it in a dish, run it through a sequencer, and you get this list of all the genetic mutations.
Right.
It's like a mechanic taking apart a car engine and just laying out every single bolt on a table.
Which, it gives you a highly detailed, comprehensive parts list. But the issue is that list is completely disconnected from where those parts actually belonged inside the engine.
You just have a pile of parts.
Exactly. You have a broken valve, maybe, but you have no idea which cylinder it actually came from.
Which is the massive blind spot we're dealing with. Traditional single-cell CRISPR screening technologies like Perturb-DBiT. It's basically like taking an entire functioning city and just dropping it into a giant blender.
That's a great way to put it.
You analyze the resulting smoothie and you say, okay, we know for a fact this city has 10,000 bricks, 50 miles of copper wire, and exactly 3 coffee shops.
But you have absolutely no idea who lived next to whom.
None at all. You don't know where the traffic jams were or which buildings were adjacent to the park.
And that loss of geography is a severe limitation, especially when we are talking about the tumor microenvironment, or the TME.
Right, the TME.
Because the tumor microenvironment is this highly structured physical ecosystem.
Yeah.
Physical location, the direct cell-to-cell interactions, those spatial cues, they define everything about how a cancer either spreads aggressively or manages to hide from the body's immune system.
So if you blend the city, you lose the map.
You lose the map entirely. And in biology, the map is very often the mechanism itself.
That's the exact problem the researchers in this Nature Biotechnology paper set out to solve. They developed large-scale, spatially resolved panoramic CRISPR screening in native tissue environments.
A very long title for a very cool tool.
It really is. And we're going to give you the complete blueprint today. We'll explore the exact mechanism of Perturb-DBiT, how it manages to capture the hidden dark matter of our genome.
Which is huge, by the way.
Oh, absolutely huge. And we'll walk through the real world, in vivo case studies, demonstrating its raw power. So to start us off, let's establish some ground rules. What exactly do we mean by a spatially resolved CRISPR screen?
Well, previously, if you wanted to see where a specific genetic edit happened inside a completely intact tissue section, your options were severely restricted.
How restricted are we talking?
You could really only look at a tiny panel of perturbations, maybe a few dozen CRISPR guides at most. Or you were limited to only looking at a specific subset of protein-coding RNAs.
Because you had to use pre-designed chemical probes.
Exactly. You had to use probes that targeted known sequences.
So you were essentially looking through a keyhole, trying to guess what the whole room looked like.
That's exactly it.
But Perturb-DBiT, which, by the way, stands for Perturbation-Compatible Deterministic Barcoding in Tissue, it rips the door off the hinges.
It really does.
I want to really dig into the mechanics here. How does it actually co-profile the CRISPR edits, like the single guide RNAs, and the whole transcriptome, the total RNA, right there on the intact tissue?
It requires overcoming this massive biological friction. Normally, messenger RNA in your cells has a naturally occurring tail made of adenine molecules.
The poly A tail.
Right, the poly A tail. Standard sequencing tech relies on that tail to grab onto the RNA. It's like a little handle.
Okay, so the machine grabs the handle.
Yes. But the CRISPR guide RNAs, the sgRNAs that we use to make the genetic edits, they do not have poly A tails.
Oh, wow. So they don't have the handle.
Nope. They are essentially invisible to standard capture methods.
Wait, if they don't have the tail, how are you pulling them out of the tissue without destroying the spatial structure?
That's where their two highly engineered strategies come in. The first is called PASEX, or polyadenylated capture.
Polyadenylated capture. Okay.
They actually do biochemistry right there on the slice of tissue. They introduce an enzyme called E. coli polymerase.
And what does that do?
It artificially synthesizes and adds these poly A tails to the CRISPR guide RNAs right there in situ.
Ah, I see. You're disguising the guide RNA to look exactly like normal messenger RNA so the sequencing system can grab both at the same time.
Exactly. You're giving it a fake handle. Once the guide RNAs have that tail, the system captures them using poly-DT primers.
Which are like molecular hooks.
Right. Molecular hooks made of thymine that naturally bind to the adenine tail. And these primers contain unique molecular identifiers, or UMIs.
Like barcodes.
Kind of like molecular serial numbers, so we can count the exact number of molecules later without accidentally double counting duplicates that get made during the sequencing process.
That's brilliant. But there's a second strategy they use alongside PBiT, right?
Yes, the DC, or direct capture method.
Direct capture.
Here, instead of chemically adding a tail, they bypass the tail requirement entirely. They use custom primers that are directly complementary to the specific scaffold sequence of the CRISPR guide RNA itself.
Oh, so they just grab it by its own unique shape.
Exactly. Standard mRNAs are captured via their normal tails, while the guide RNAs are captured directly by their unique molecular signature.
Okay, I'm with you on the capture part, but here's my biggest question. You've grabbed the RNA off a glass slide, but how do you know where on the slide it came from?
Right, the geography part.
Yeah. If you're washing enzymes over a tissue slice, doesn't the RNA just float away? How do you map it?
Well, the tissue is fixed and permeabilized, so the molecules stay locked in place, but they're still accessible to the chemicals.
Okay.
And to map them, they use a PDMS microfluidic device. To visualize this, I want you to imagine placing a microscopic, invisible chessboard directly onto a thin slice of tissue.
An invisible chessboard. Okay, I'm picturing it.
This microfluidic chip has microscopic channels running through it. First, they lay down a set of horizontal channels across the tissue.
Going left to right.
Right. And through these horizontal channels, they flow in a specific 8-base pair genetic barcode. We'll call this the AI barcode, numbered 1 through 100.
So you've essentially painted 100 microscopic horizontal stripes of barcodes across the tissue slice.
Correct. Then they remove that device and carefully place a second microfluidic device on top. But this time, the channels run vertically.
Top to bottom.
Exactly. They flow in a second set of barcodes, the BJ barcodes, also numbered 1 through 100. But crucially, this time, they flow them in alongside an enzyme called T4 DNA ligase.
The ligase acts like molecular glue, right?
Yes, exactly like glue. So where the horizontal stripe and the vertical stripe cross.
The ligase permanently glues those two barcodes together.
You've got it. That geometric intersection creates a completely unique 2D spatial coordinate.
So 100 times 100 gives you a grid of 10,000 distinct spots across the tissue.
Right, and each one is about 50 microns wide. So when we eventually sequence the RNA, we just read the combined AIBJ barcode, and we know exactly which microscopic square on our chessboard that specific RNA molecule came from.
That is just wild.
Ah.
Okay, before we move to the clinical applications, let's just make sure we're fully grounded here with a little mini summary.
Sounds good.
We've basically moved from the old blender method to laying down a microscopic GPS grid over an intact piece of tissue.
Right.
And using either enzymatic tails, the PAC method, or direct capture, we can read both the genetic blueprint and the specific CRISPR edit simultaneously.
And we know where it came from.
Exactly. We know exactly which 50-micron neighborhood it came from because of those intersecting horizontal and vertical barcodes.
That's the perfect synthesis.
I want to throw a question out to you listening right now. Think about your own daily commute for a second. If a city planner only looked at the plumbing of your specific house, could they possibly solve a rush hour traffic jam 5 streets down?
Obviously not.
Of course not. You need the spatial context. You need the map to understand the whole system. So just to give a quick 5-minute recap for everyone keeping track. Bullet 1, traditional CRISPR loses spatial data, and older spatial CRISPR was just too limited in scale.
Right, the keyhole problem.
Bullet 2, Perturb-DBiT fixes this by mapping total RNA and CRISPR guides simultaneously on intact tissue.
Yes.
And bullet 3, it uses this microfluidic coordinate system with specific AI and BJ barcodes to track exactly where every single read came from.
Spot on. And, you know, having that spatial context is what they tested next. Because having the mechanism is great on paper, but biology is notoriously messy.
Oh, extremely messy.
So they had to prove this GPS tracker for genetic edits actually works in a living, breathing system.
Right. So let's talk about that first in vivo proof of concept. They started with a spleen model, right?
Yes, they did. They used a SMARTA mouse model.
SMARTA?
Yeah. These mice are genetically engineered, so they're CD4 plus T cells, the cells that kind of quarterback the immune response, all recognize a specific virus.
Okay, so they have a very focused immune system.
Exactly. And they infected the mouse with LCMV, or lymphocytic choriomeningitis virus, to kick off a massive immune reaction.
So they basically created an active immune battlefield in the spleen, and then they introduced a small library of CRISPR guide RNAs into those T cells.
Right.
What did they actually see when they mapped it with Perturb-DBiT?
Well, they analyzed the spleen tissue 6 days post-infection, and the results were remarkably clear. The non-targeting control guides.
Wait, what are those?
Those are the T cells that received a blank CRISPR edit, so they still had fully functioning genes.
Ah, okay. The controls.
Right. Those control guides aligned perfectly with the B cell zone of the spleen.
Which proves what exactly?
It proves the CD4 plus T cells were migrating and physically interacting with the B cells exactly where they're supposed to be during a normal viral response.
Oh, wow. So the microfluidic GPS system successfully found them in the correct neighborhood.
It functioned beautifully.
But I mean, a small library in a spleen is one thing. They scaled it up after that, right?
Yeah. They immediately stepped it up to a medium-sized library in a much more complex environment, a liver tumor.
A liver tumor? That sounds much messier.
Definitely. They used an autochthonous liver tumor model.
Autochthonous. Explain that term for us.
Autochthonous means the tumor naturally developed in the mouse's liver over time from its own cells, rather than just being a lab-grown cell line that researchers injected into the animal.
So it perfectly mimics natural disease progression.
Yes. And they introduced a library of 288 different CRISPR guides into this natural liver tumor.
288 different edits happening at once. And did the GPS grid find them?
It did. Perturb-DBiT successfully mapped up to 265 out of those 288 unique guide RNAs spatially across the liver tissue.
Okay, I have to play the skeptic here, though.
Go for it.
Finding 265 out of 288 sounds great. But if you're taking 50-micron spot readings, how do you know the spatial reading isn't just noise? How do we actually know this is as accurate as the old blender method?
That is the exact right question to ask, and it brings us to the evidence level of this paper, because they knew they had to prove that.
Right.
They compared the Perturb-DBiT data directly against EMIS. EPS stands for Molecular Inversion Probe Sequencing. It is the gold standard for traditional non-spatial pooled sequencing.
Okay, the gold standard.
And they ran an ROC curve analysis to see how well their new spatial method distinguished true biological signals from just random background noise.
And what was the verdict? How did it score?
The AUC, or area under the curve, was 0.724.
Good.
In the world of high-throughput spatial diagnostics, an AUC of 0.724 against a bulk sequencing standard proves high fidelity.
Okay.
It means Perturb-DBiT isn't just generating pretty maps, you know? Its 50-micron captures are highly concordant with the established biological truth. It captures the exact same reality, just with the added dimension of space.
Let's pause and digest this because the implications here are just huge. Here's a mini summary of this section.
Yeah.
The technology was validated across different scales, from a small viral response in a spleen all the way up to tracking hundreds of genetic knockouts in a naturally occurring liver tumor. And it proved to be mathematically just as reliable as traditional non-spatial methods, but it gave researchers the actual physical map of the organ.
It gives you the where, not just the what.
Exactly. So I want to ask you, listening again, if you could map the exact physical location of a specific gene mutation in any organ of your body, where would you look first?
It really changes how we view illness.
It fundamentally does. But I know we promised to make a case today. We've talked about a few hundred guides. Now I want to talk about the genome scale test.
The big one.
Over 80,000 edits.
This was the ultimate stress test for the system. They applied Perturb-DBiT to a human colorectal cancer model, specifically the HT29 cell line.
And what does that cell line do?
It aggressively metastasizes to the lung.
Yeah.
And they didn't hold back here. They used a massive genome-scale library containing over 77,000 to 80,000 different guide RNAs.
I have to stop you there. 80,000 different genetic edits happening in a single model.
Yeah, 80,000.
How do you even begin to process that visually? That sounds like a bioinformatics nightmare.
Oh, it requires immense computational power. To solve this, they introduced a computational framework called iStar.
iStar. Like a bridge.
Yes, exactly like a bridge.
Right.
It takes the genomic data from those 50-micron Perturb-DBiT barcode spots, and it computationally fuses it with super high-resolution histology images.
So the physical sub-micron microscope pictures of the tissue.
Right. By mathematically mapping the genetic data onto the visual textures of the cells, iStar creates these super-resolved near single-cell maps of the tumors.
So the computer algorithm basically helps fill in the visual gaps between the 50-micron grid coordinates.
Correct.
And when they looked at those high-res maps, what did they actually find?
They found the specific mutations that were driving the human cancer to colonize the mouse lung. The top hits were all tumor suppressor genes.
Genes that usually stop cancer.
Right. When the CRISPR guides snipped these specific genes out, the cancer cells suddenly thrived and spread. The major genes they identified were SEMA6B, MT1E, SMNA4, and ASAH2.
Okay, those are a lot of letters and numbers. To understand why this is profound, we have to look at what those genes actually do. Take SEMA6B, for example.
Sure.
Semaphorins are generally involved in cell signaling, acting as boundary markers. They act as a sort of brake pedal for cell movement.
Exactly. So when the CRISPR guide knocked out SEMA6B, the cancer cells literally lost their brake pedal.
They gained the ability to physically migrate and colonize the lung tissue.
Yes. And the crazy part is, we didn't just guess that this happened. Perturb-DBiT allowed the researchers to literally watch it unfold across the tissue.
Which brings us to pseudotime, which might genuinely be my favorite concept in this entire paper.
It's a brilliant way to analyze the data. They used an algorithm called monocle 3 to map the evolutionary trajectory of these tumors directly on the tissue.
Okay, the best way to visualize pseudotime, and I love this analogy, is to think of a time lapse of a city expanding.
Okay.
But you are only allowed to look at a single still photograph.
But you don't have a video.
Exactly. If you look at the photo and you see a cluster of brand new skyscrapers next to a neighborhood of half-built high-rises, which is next to an empty lot with bulldozers, you can logically infer the timeline of how the city evolved just by looking at the physical states sitting next to each other.
That is a perfect analogy. By analyzing the transcriptional pseudotime across the lung tissue, Monocle 3 showed how specific perturbations, particularly the knockouts of SEMA6B and MT1E, literally forced the tumor to biologically evolve.
So you could see it differentiating as it expanded outward.
Yes. You are literally watching the genetic driving forces of cancer metastasis unfold geographically in a single snapshot.
That's incredible.
Yeah.
But the authors didn't just trust their own novel tech, right? They cross-referenced this against existing platforms.
They did. They validated these spatial findings against the 10x Xenium platform.
What's Xenium?
It's a state-of-the-art in situ imaging system. And Xenium completely confirmed the spatial locations of these top tumor-driving hits.
Nice.
But there is a massive, massive distinction to make here.
What's that?
Xenium is fantastic, but it relies on targeted probe panels. You have to tell the machine exactly which genes to look for in advance.
Oh. It's the difference between searching a dark city for a suspect with a narrow flashlight versus just turning on the sun and seeing everything at once.
Exactly. Because Perturb-DBiT captures unbiased total RNA, it revealed a vastly broader, wildly diverse clonal landscape of the tumor that the highly targeted Xenium panel simply couldn't see.
Because Xenium wasn't programmed to look for it.
Right. Perturb-DBiT removes human bias from the discovery process.
So just to lock this massive section in with a mini summary, by applying this tech to an 80,000-plus guide library, researchers discovered the exact genetic brake pedals like SEMA6B and S1000A4 that human cancer cells disabled to colonize lungs.
Yes.
And using algorithms like iStar and Monocle 3, they tracked the physical evolutionary paths of these cells across the tissue.
Which raises an incredible prospect for the listener to think about. We just saw how a single genetic tweak changes how a tumor physically spreads.
So if you are a pharmacologist, how does this change the way you design cancer drugs in the future?
You're no longer just targeting a mutation. You are targeting the geography of the mutation.
It's mind-blowing. Let's do a quick 10-minute recap for everyone. Bullet 1. Perturb-DBiT successfully mapped a massive 80,000-plus guide library in vivo.
A huge achievement on its own.
Bullet 2. The iStar algorithm helped create near single-cell resolution maps of these tumors by fusing the data with histology images.
Right.
And bullet 3. Pseudotime analysis proved that we can see exactly how tumors evolve spatially based on specific genetic mutations.
Spot on.
And that naturally leads us to the final major frontier explored in this paper. Because as incredible as mapping 80,000 genes is, traditional biology has had a massive blind spot.
A very large blind spot.
We've basically been ignoring the dark matter of the genome.
Yeah, when biologists talk about RNA, they're almost always referring to mRNA messenger RNA.
The stuff that makes proteins.
Exactly. This is the RNA that codes for proteins. But that protein-coding mRNA is only a tiny fraction of the genetic story. There is an entire universe of non-coding RNAs.
Like what?
like long non-coding RNAs or lncRNAs, microRNAs, and transfer RNAs. Traditional probe-based spatial methods almost entirely miss this dark matter.
Because those probes are only designed to attach to known protein coding sequences.
Right. They literally don't have the tools to see it.
And this is why Perturb-DBiT is such a paradigm shift.
Yeah.
Because it uses poly-A capture and direct capture, it grabs total RNA without bias.
It sees everything.
But why do we care about the non-coding RNA?
Yeah.
What does this dark matter actually do in the cell?
We're discovering that these non-coding RNAs act as highly complex post-transcriptional regulators and metabolic switches.
Meaning they control things after the blueprint is read.
Right. They can silence other genes, alter cellular metabolism, and radically change how a cell behaves without ever actually producing a single protein themselves.
Okay, let's use a mnemonic to really burn this into our memory. Think of standard messenger RNA as the blueprint for a building.
Okay.
It tells you where the walls and windows go, but the non-coding RNAs, the dark matters, they are the construction managers.
I like that.
They don't provide the raw materials, but they are the ones on the ground deciding exactly when the building gets built, where it is allowed to go, and how fast the workers move.
Yes, that's exactly it.
Most older technologies only let you read the blueprint in an empty office. But Perturb-DBiT allows us to watch the construction managers barking orders on the actual job site.
The construction manager analogy is incredibly accurate, especially when we look at their final case study. They wanted to see these managers at work in the immune system.
Okay.
So they use a syngeneic mouse model.
Syngeneic.
Syngeneic means the mouse had a fully functioning native immune system. They wanted to map the tumor immune microenvironment. And to do this, they combined Perturb-DBiT with another technology called Codex spatial phenotyping.
Okay, so what happens when you watch the tumor interact with a fully armed immune system?
You see exactly how the tumor fights back. They found specific spatial clusters within the tumor that were heavily manipulating the immune response using non-coding RNA and specific genetic pathways.
Wow. Do we have specific examples?
Yeah, for instance, they found one tumor cluster that was enriched with a PAX8 perturbation and another completely distinct physical cluster with a ZC3H12A perturbation.
And what were those specific clusters actually doing?
These genetic knockouts actively modulated the physical architecture of the tumor to promote immune suppression. They were literally pulling in very specific immune cells like dendritic cells and tumor-associated macrophages and hijacking them.
Hijacking the immune cell.
Yes. The tumor was creating localized immunosuppressive pockets to ensure its own survival.
It's like the tumor is editing the zoning laws of the city to build its own private fortress, bringing in corrupt cops to guard the perimeter.
That's exactly what it's doing.
It's terrifying to visualize, but mapping it is the first step to dismantling it. So here's our final mini summary. By capturing total RNA, Perturb-DBiT maps the invisible dark matter of our genome, revealing exactly how tumors manipulate their local immune environment to survive.
It reveals the full regulatory landscape, completely unbiased.
And so I ask you, the listener, knowing that the dark matter of our DNA plays such a massive role in how cells physically organize and defend themselves, what other mysteries of the human body do you think this dark matter is hiding? It feels like we're just scratching the surface here.
We truly are. It's just the beginning.
So as we wrap up this deep dive, let's bring it all into focus. This paper fundamentally proves that Perturb-DBiT successfully marries the massive genome-wide scale of pooled CRISPR screens with the geographical precision of spatial transcriptomics.
Right.
And it does it totally unbiased, without relying on limited targeted panels. We can now see the blueprint, the managers, and the geography all at once.
It is a massive technological leap forward. However, it is really important to objectively address the current limitations that the authors themselves note in the paper.
Yes, let's be impartial here. What are the drawbacks?
First, the technology is constrained by the actual physical area of the tissue being sampled. If you use a massive library of 80,000 guides, not every single one of those perturbed cells is going to happen to make it into the specific tiny tissue slice you place on your microfluidic chip.
Oh, right, because you're only slicing a thin piece of the tumor.
Exactly. You are getting a highly dense sample of the library, but not the entire thing in one single pass.
Which means to get the entire 80,000 guide picture, you'd have to sequence dozens, maybe hundreds of consecutive tissue slices, which is incredibly resource intensive.
Extremely resource intensive. And the second major limitation is resolution. Right now, those grid coordinates are 50 micron spots.
Which is small, but
It is incredibly detailed for capturing total RNA, but it is not true, absolute single-cell resolution. There are still multiple cells packed into a 50-micron spot.
I see.
Achieving true single-cell spatial resolution across the total transcriptome is definitely the next major hurdle for this field.
But even with those limitations, I mean, the ability to see the geographic impact of a genetic edit on both the coding and non-coding RNA is just staggering.
It changes everything.
Which leaves me with a final thought for you. We just spent this entire time talking about cancer models, but I want you to think bigger.
Much bigger.
If we can map exactly how a single genetic edit reshapes a physical neighborhood of cells and alters the dark matter of the genome, what happens when we apply this to the aging brain?
Oh, wow.
Could we use Perturb-DBiT to map the exact genetic tweaks needed to stop neurons from degenerating, block by block, cell by cell, in diseases like Alzheimer's?
That is the exact kind of systemic, complex question this technology finally gives us the vocabulary to ask.
It's an incredible thought. Keep questioning the blueprints of life, everyone. Thank you for joining us on this deep dive into the source material. We'll catch you next time.